Good. So, lecture number 12. So, as you might guess, we have already went for quite a long path through the organic chemistries, through the biochemistry, and this is already the 12th lecture. We have still 3 lectures in the future, and I guess they will be more inclined towards neurobiology. It may be a little bit, I don't know, maybe a little bit controversial choice on my side, but I hope you will like them as well. But today we have clearly a biochemical lecture on the biochemistry of the liver. The thing is that the biochemistry of the liver is so vast that I don't think anyone can fit that into 1 lecture or even 2 lectures. Um, there is some enormous amount of known biochemical pathways that are going in the liver. So as far as I was concerned, it's something around 500 known biochemical pathways. That's of course not including unknown biochemical pathways that we may not know. So there's a really large field to study and to get through. Moreover, even from the known biochemical pathways, and even taking the few of them that are always included into classical biochemical courses, we can't get through all of them in 1 lecture once again. So there are certain pathways that I will leave, let's say, on the side road, and just few of them that differ the most from the pathways we have already considered I will take into this lecture. So just for you to know, there is much more to study about biochemistry of the liver. So I will be mostly focused on the biochemical pathways regarding some metabolism of lipids, and a little bit more regarding metabolism of the nitrogen. I'm leaving aside some notes regarding glycogen, so just for you to notice, there are certain specific things regarding glycogen metabolism in the liver, and we use it as some sort of battery, so that glycogen in the liver can be transformed into glucose that is transported into the blood and used to, well, with different organs. And there's you can use that sort of supply for, I guess, something like 20 hours, so there's a lot of energy stored in the liver. Moreover, there are some pathways like cytochrome P450 pathways that clean up different mess, and these are pretty interesting pathways as well. They metabolize different chemicals that we don't want to have in our organism, or we want to get rid of them somehow. There are pretty interesting things regarding these cytochromes, so they go with different numbers, and for example, cytochrome, they are called cytochromes 450 or CYP 450, but they often come with numbers like cytochrome CYP1A2 is metabolizing, for example, caffeine. So if you drink a lot of caffeine, that's the cytochrome that tries to get rid of that from your organism. And interesting point to notice that if you like, for example, grapefruit juice, it contains a chemical called, I guess, naringenin or naringinin. Not sure how to read that, and this chemical is inhibiting these exactly cytochrome. And I even tried at some point, and even tried to mix coffee with this sort of juice to get some prolonged effect of the caffeine. I should tell you that results, well, were not much promising, but sometime later I found an article that in some sense that works well, in a sense that you can measure the effect if you are measuring certain concentrations of that chemical, so on, but the effect is too small to be felt, so you will not feel anything, but if you measure everything, you can find certain effects, so it is, and so on. Of course, certain chemicals like alcohol dehydrogenase, it should be considered, and many other things, but I'm a little bit avoiding that for a while. Well, if we get into that let's say tricky question for you, what is the enzyme responsible for hangover? Who knows? Any ideas? Nope. I guess we have seen these enzymes somewhere in our lectures. I just can't remember where it was. There are two enzymes, alcohol dehydrogenase and aldehyde dehydrogenase. So alcohol dehydrogenase transforms ethanol into acetaldehyde, and the acetaldehyde, that's basically the chemical that makes you feel bad. So here it is. What's more interesting, if it somehow happens that human drinks, for example, methanol, so you know that drinking methanol is, well, that's deadly poison, so better never do that. But the thing is that you might be wondering why, if it looks pretty similar to the ethanol. And the thing is that in the liver it is metabolized to the formaldehyde, and formaldehyde is the exact chemical that does the damage. So methanol on its own is pretty harmless, while if it gets through the alcohol dehydrogenase to the formaldehyde, it starts doing a whole lot of nasty things and destroying everything around, so the consequences are pretty dire. And one of the ways to treat this sort of poisoning with the methanol is trying to inhibit alcohol dehydrogenase to keep methanol in form of methanol and not let it to get to the formaldehyde form. Okay, that was a large step aside. Let's get to what we have for today. So if we are talking about liver, you know that I always like to position ourselves in, well, where we are, so where are the processes we are talking about. So processes in the liver, so here you see the liver of a human. Of course, many other living organisms have liver as well, but you might guess that that humans are the organisms that we are concerned mostly, so that's the most interesting part of the biochemistry. Here you see in 3D, so you can see how that's positioned with the respect to the rib cage, and if you have ever been to a doctor, let's say, and he's trying to palpate something, so he may be trying to touch the liver and to get certain idea regarding its form, size, maybe what's going on with the gallbladder, and basically that gives some hints for providing certain diagnosis or something like that. So here are few more schemes. So here you see on the left you see blood circulation. So here you see the portal vein. Portal vein takes up many different chemicals that are results of the digestion of food and brings all that things to the liver. So if you have ever studied pharmacology, you may have noticed that when it gets to some sort of pills, and you are performing certain calculations, there is the thing called bioavailability that is basically result of these first pass through the liver when all that drugs are coming through the portal vein to the liver, and then from the liver they will be exported basically to the blood and can be used by different organs. So of course liver performs a lot of processing on that chemicals, and she's trying to remove anything that may be harmful for the organism, so not to let it basically to go into the bloodstream and affect something else, and maybe some, well, and different, all the different processes are performed. Here on the right you see once again our digestive system, so in its full glory. So here you see the esophagus, here are all the different sphincters and parts of the stomach and so on and so forth. What's interesting here, so here you see the liver, this is the gallbladder, and here you see this tiny tube that is going right here and ends up with the sphincter of Oddi. And here you might guess that bile salts are basically transported to our intestine, so to the small intestine, and they are performing the work of emulsifying the different lipids. So they should be emulsified to be transported then to the bloodstream and then processed into something useful for our organism. So here you see that point of entrance. Here is another view. So once again you see the anterior view of the liver. So here you see the diaphragm, here you see the liver, once again the gallbladder. But what's interesting here, here is some enlarged view of a small portion of the liver. So you see that a liver is constructed from these sort of small hexagons, and here you see interesting parts of that hexagons. So here is so-called portal triad that basically contains a portal venule, a portal arteriole, and a bile duct. So bile is produced into that bile duct, and blood is supplied through that portal venule and arteriole into these into this small part of the liver. So here is a view from another side, like some sort of cross section. Here you see that. So here's once again the bile ductule and portal venule and hepatic arteriole, and here's how that looks like. Here is a 3D view, so once again you can look in 3D how that looks like. So here is our hexagon. One part of this hexagon was cut out, so you can look a little bit inside how that looks like, and that should give you some sort of idea regarding the structure of the liver. So here is even more enlarged structure of the liver. So what do you see here? You see this central vein, and here you see this triad. So the portal triad, let me show you. So here was that central vein, and here was the portal triad, and this is portal triad, and this is, and so the whole hexagon. So here you see them. Here you see different cells, different spaces, and so on. So here are some sort of, let's say, Kupfer cells. These are sort of macrophages, but these macrophages don't wander around as usual, because you probably know that macrophages, they have tendency to walk here and there and so on, and it just sits here and eats everything that looks more or less suspicious and may be harmful for the organism. So here you see these this sort of structure. On the left, you might see some sort of microscopic structure, so how that looks under the microscopy. So you see some hepatocytes, you see the portal vein, how that looks in here, and you see the bile duct and some hepatic arteriole, and that's how it looks like. Well, I never was strong with all that microscopy, so so that's really a little bit miracle for me that people are able to recognize all that stuff. But they certainly are able. So once again, some more nice 3D pictures for you, just to get a grasp of what is the microstructure of the liver. So here you see that, and here is a schematic view. So from the of all the different cells, and here you see the space of DC and the sinusoidal lumen. That's what have you seen in the in here. So here you have seen the space of DC, and here you see the sinusoidal lumen. So we are considering this sort of portion of the wall, so this sort of wall. So here you can see it, you see these sort of tight junctions. So tight junctions are tightly connecting cells, and most chemicals cannot diffuse through that, so it's really, they are really tight. And here is the structure. Regarding synthesis of all that bile acids and so on, I guess we will get, maybe not today but tomorrow, we will talk a little bit about the synthesis of the bile acids. And here is some foreshadowing regarding their circulation. So they are synthesized, and how the turnover of all that biochemistry, how it's going on. So here you see the hepatocytes, that tight junctions. So that comes from the previous image. Yeah, so that's that wall of cells. Cells are connected with tight junctions, but they may have these sort of canaliculi between them, and they can actively secrete something into these sort of canaliculi, and that may be that may include, of course, bile acids, and that may include some interesting lipids like phosphatidylcholine, or maybe there is some conjugated bilirubin and some xenobiotics that we may want to get rid of. So here you see a little bit more of the anatomy. So here you see the common bile duct, the gallbladder, the liver on its own is not shown here, so it should be near the gallbladder, and here you see just some hepatic ducts from the liver that are delivering these sort of bile acids, and they are then going into the duodenum. So that's that's in here, and that's a part of small intestine. So small intestine, it has like some closer part closer to the stomach, that is called duodenum, and then there is jejunum that is then in closer to the large intestine. So, and here you see the circulation of the bile acids. So they come through the sphincter of Oddi into the small intestine, and then they are reabsorbed. So, and these reuptake of the bile acids may return them back to the liver, and then they will be secreted once again and again and again. Here are some different pathways, so a little bit may circulate through the bloodstream, and they may be measured there, and there is a little bit of passive reuptake. So relative amounts of bile acids in different body pools and the enterohepatic circulation they are indicated with the widths of these green lines, and this figure basically illustrates that the majority of the bile acid pool circulates between the liver, gallbladder, and small intestine. So it's not in a systemic circulation; it's there. Good. So now let's get a little bit closer to something that has to do with biochemistry. I hope we have more or less we have positioned ourselves, so we know what organ are we talking about, and now we will be dealing with the biochemistry of the liver cells. So what can they do for us, and what's so interesting for us in all that cells. And and here we start lipid catabolism. So we are getting energy from all that lipids. Moreover, we use them for as some sort of structural components and signaling molecules, and even for protein modification. So first of all, we can store 3-acyl glycerols or different neutral fats, and these are, as you might guess from the name, these are fatty acids that are connected with the glycerol, and they are stored in the adipose tissue, or its cells are called adipocytes. And depending on our energy needs, we may mobilize these sort of fatty acids, and we may oxidate them for the ATP production. There are certain caveats regarding that. We can mobilize them and produce some ATP, and moreover, that's like a primary energy source at rest or during moderate exercise. But there's an interesting caveat that I will show you a bit later. We cannot convert them back to the glucose. We can get only to the acetyl-CoA. That may seem a small problem, but there are certain organs in the organism that need glucose for their functioning. So you can't give them acetyl-CoA. You cannot just transport that acetyl-CoA into the bloodstream or something like that, and there is a bit of a problem. And the problem is that these organs include our brain. So there are certain workarounds to keep our brain alive in case we have troubles with getting glucose, and while all the rest are getting some energy from these sort of storage, from fats that are stored in the adipose tissue. But you should know that there is this sort of caveat, so we cannot transform that back to the glucose, which probably might have been the most perfect way of using this sort of storage. Well, but anyway, that works. What else? We need fatty acids as building blocks. So you might guess that all our all our cells, all their membranes, they are they are created as some sort of bilayer from lipids, and first of all from phospholipids, and basically that's all synthesized from the sort of acids. And what else? Some proteins may be covalently acylated, so like adding some fatty acids to the protein to modify it, maybe to help it to anchor to the membrane and to let it still be in the membrane. And we are using some fatty acids derivatives as hormones or intracellular messengers, so they may be used for some sort of signaling. So how do we get them? Mostly we get them from eating something, from our diet. So we are ingesting some fats. Here you see once again, so whatever we have eaten has passed our stomach. So here you see the gallbladder, the sphincter of Oddi. And here in the small intestine, the bile salts that were produced by the liver in that circulation that I showed you, that green circle, they emulsify dietary fats in the small intestine and are forming some sort of mixed micelles. So here they are. So here you see the small intestine, and here you see triacylglycerols and what is basically happening. The intestinal lipases, they are degrading triacylglycerols, removing glycerol from the fatty acids. Fatty acids and some other breakdown products, they are taken up by the intestinal mucosa and are converted into triacylglycerols back. So we are first degrading everything, then we are choosing what we need and transporting that, then we are collecting things together to make something more handy for us for the transportation. And this sort of transportation, so these triacylglycerols, they are incorporated with cholesterol and certain apolipoproteins like ApoC2, and that transforms them into so-called chylomicrons. And these chylomicrons, they can move through the lymphatic system and bloodstream to different tissues. So we are transporting this way all the different fatty acids to different tissues where they may use them for something. So either for storage or maybe for getting immediate some energy. So whatever they need. Here you see the lipoprotein lipase that is activated by the ApoC2 in the capillary, and that converts once again triacylglycerols to fatty acids and to glycerol. Fatty acids, they can enter these cells. They can enter cells because basically they are hydrophobic, so it's easy for them, and then they may be or oxidized if we need energy right now and used in the Krebs cycle, so they will be transformed into acetyl-CoA, and that acetyl-CoA will go to the Krebs cycle, or they may be once again reesterized and transformed into triacylglycerols that may be stored in that tissue, so for some future usage. Maybe we will need them. Here is a little bit regarding that sort of bile salts. So here you see how that looks like. So this part is added in the liver, and this OH group, then the other OH group may be removed by gut bacteria, and that transforms cholic acid into deoxycholic, or chenodeoxycholic into letha-cholic acid. So there are certain types of that bile acids and bile salts in normal conditions, but here is some sort of general formula that more or less tries to encapsulates them. So you might guess this part, so it comes from the cholesterol, so they are pretty similar to cholesterol. Here you see the mechanism of getting that sort of fatty acid on the micro level. So here is the stomach, once again lumen of small intestine, and here you see the villi and intestinal mucosa, and here is the epithelial cells of that villi, and the microvilli or brush border, and all the rest things. And these are fatty acids. They are transported through these sort of villi. So each of the villi in the small intestine consists of a layer of epithelial cells and the core of capillaries and some connective tissue. The outside, the apical face of each epithelial cell is covered by smaller projections called microvilli. So here you see them. And here is basically the formula for the triacylglycerols. So here you might see the part that comes from the glycerol. If after these O's I will have just simple H's, so just hydrogens, that will be just a glycerol. So that's an alcohol that has 3 OH groups, so nothing very special, well, pretty simple structure. And here you have these sort of ester bonds that that were created by reacting that OH group with the group of the acid. So of 3 different fatty acids. And here you see the action of the pancreatic lipases that are trying to make something out of it, to remove some of the fatty acids from the glycerol, and here you see what may happen. So these sort of lipases may transform that into beta monoacylglycerol, so removing 2 of them, or into diacylglycerol, removing one of the fatty acids. So that's basically what's happening. And here you see the back process of synthesis. So here you see 2 fatty acids. They are coming with 2 CoA, so 2 molecules of the coenzyme A. 2 fatty acyl CoA are formed. So don't mix up. So acyl CoA and acetyl CoA. So acetyl CoA, that it means that CoA is attached to the acetyl group. So just 2 carbon atoms. Acyl CoA means it is attached to a fatty acid. Fatty acids are pretty long, typically something like 16 carbon atoms, maybe more, maybe less, but that's a typical number. So that's for you to know. And these 2 fatty acyl CoA, so they come with the monoacyl glycerol and can be collected together once again to this triacylglycerol that will be incorporated into the chylomicron and then will be exported into the lymphatic duct and maybe into the bloodstream and then to the organs that may need these sort of these sort of molecules. Here is the structure of the chylomicron, and here you see a triacylglycerol once again. So here are 3 different fatty acids, so don't think they should be all equivalent. They may be pretty different. And here you see different apolipoproteins like C2, B48, T3, and triacylglycerols and cholesterol esters. They are inside of this thing, and the outside, that's a membrane formed with phospholipids. And the chylomicrons, they enter the lymph, then blood. The APO seek to activate lipoprotein lipase, and once again triacylglycerols can be deconstructed into fatty acids and glycerol, and then fatty acids may get to some sort of oxidation, for example in muscles, or it can undergo some re-esterification and storage, for example in adipose tissue. The remnants, they may be returned to the liver and uptaking via the ApoE mediated endocytosis. So the surface is the layer of phospholipids which had groups that are facing the aqueous phase. You might guess basically why. So water is is polar, so it likes something polar. So if we are creating a molecule that has some charge somewhere, so it will try to turn that charge to the water. So that gives some sort of minimum of the energy. So that's the reason why do we have a lipid bilayer as a membrane in our cells, because we have water on both sides, and we are trying to let's say turn the polar heads towards the water. In here we have a little bit different situation because we are collecting triacylglycerols inside of this chylomicron. They are hydrophobic, they don't like water so much, so we are okay with not bilayer but just with one layer of phospholipids, because the inside that fat tails, they will feel pretty comfortable with the inside. There is no water there, so that's all hydrophobic, while the polar heads will be pretty happy with water outside because water is polar. The apolipoproteins that protrude from the surface, they act as signals in the uptake and the metabolism of chylomicron contents, and their diameter ranges from 100 to 500 nanometers. They are pretty abundant. I haven't seen that myself, but as far as I heard, if someone is doing some sort of analysis of the lymph after a good fat meal, that lymph will look even, people say it looks even white, because there is a lot of all that chylomicrons and all that fatty acids are transported from the liver, and that can be even seen by naked eye. So the color changed, and that comes after a good meal that contained a lot of different fatty acids and so on. Just a notice regarding apolipoproteins and lipoproteins. They help with the transport of dietary and endogenous lipids. We are synthesizing some lipids on our own. We will get to that tomorrow. I guess I will show you some pathway how do we do that. So if we need, we can synthesize at least some lipids. So apolipoproteins, they are lipid binding proteins that transport triacylglycerols, phospholipids, cholesterol, and cholesterol esters. Apo means lipid-free protein. Combined with lipids, they form lipoproteins. So there is some sort of hydrophobic core, so triacylglycerols and cholesterol esters, and hydrophilic shell that contains phospholipids and apolipoproteins. So that's what you have seen in these slides. So hydrophobic core and hydrophilic shell. And they perform some sort of structural support. ApoB48, ApoB100. So here you see ApoB48 incorporated in this thing, so there may be some different proteins that are helping to support the structure. Some are enzyme cofactors like ApoC2, while other are receptor ligands, so they are basically telling the cells what is this thing floating around, and are activating certain signaling cascades that will tell what to do with that. Do they want to uptake that thing inside, or just leave it as it is? So that depends. And some transfer proteins. Good. What else? Here are some classes of lipoproteins by density. These are the collections of the of different proteins and some lipids. The least dense and the largest are chylomicrons that we have been talking about, though that are transporting all that stuff that we have ingested to different tissues. and the remnants are uptaken in the liver via the ApoE receptors. They are rich in triglycerides, triacylglycerols. There are three more classes that are called very low density lipoproteins, so VLDL, IDL intermediate density, LDL low density, and HDL high density. So they are a little bit different in their origin and fates, but so VLDL, they originate in liver, and they basically deliver the endogenous triacylglycerols to the tissues. So when we get tomorrow to the biochemical process of synthesizing some lipids, of course, if we have synthesized that lipids, we need to transport them somehow to different tissues. So liver is not synthesizing them for itself. It should give it away to different tissues, and it is transported in these sort of very low density lipoproteins. And these are basically, they originate IDL and LDL, they originate as the results of some sort of catabolism, like VLDL are catabolized to the IDL, and IDL, they may be catabolized further, and then they give you LDL, and these LDL, they basically deliver some cholesterol to peripheral tissues, and there is some, they have certain clinical meaning, like they are called like bad cholesterol, so they may indicate some risk of atherosclerosis, so there may be. And there is HDL, so high density lipoprotein. They originate in liver and intestine, and they have the function of the reverse cholesterol transport, so they are transporting cholesterol from the peripheral tissues back to the liver in case we have too much of the cholesterol, and some of it should be somehow metabolized and excreted. So and they are, in clinical practice, so they can be say called a good cholesterol, so they are indicators of good cholesterol. Okay, here is an example of the mobilization of the triacylglycerols stored in the adipose tissue. So we have taking all the triacylglycerols. We may have stored them somewhere. Well, that's tissue, that's adipose tissue, and here you see on this image where we have different parts of that adipose tissue. It's almost everywhere. So you see we have that even in the bone marrow. So in some places in organism we have more of that, in some we have less of that, but we have adipose tissue almost everywhere. And here is the example of the metabolism when we want to mobilize that triacylglycerols and use them for getting some energy. So here you see a glucagon receptor that basically starts that sort of cascade. I hope you remember two hormones like insulin and glucagon, and insulin is saying to your tissue that we have just eaten something, so guys, we have a lot of different useful stuff in the bloodstream, take it and store it. So take it and store it. While glucagon says that we haven't had meal for some time, but we need to do some work. So guys, if you have some storages of energy, maybe it's time to use them. And that's basically what's happening in here. So glucagon tells you that you have to mobilize some storages of the energy. Here you see a receptor. Here's the tricky question for you. Can you guess the type of this receptor? Should be pretty simple. No? No one can guess? At least here are two hints that should be pretty evident for you. So one is a slight hint, the other one that's incredible hint. So a slight hint, that's adenylyl cyclase. Yes, GPCR. So G protein coupled receptor. Absolutely correct. Yes, yes. That's it. So here you see though a small hint that's adenylyl cyclase. So you might have remembered that the adenylyl cyclase is activated with GPCRs. The other, just I don't know, some looming hint, that's this small portion GS. So you may have guessed that it's all about the G protein. So, and if you have G protein, of course, the receptor will be G protein coupled. Okay, so now what's happening here? Of course, the adenylyl cyclase synthesizes some cyclic AMP, a second messenger. It activates protein kinase A, and this protein kinase A starts doing different things, like phosphorylating the hormone-sensitive lipase. So here it is. Or it may start phosphorylating the perilipin. So here you see the perilipin, here it is phosphorylated, here this perilipin is connected to CGI51. So it may be released from the CGI51 and phosphorylated, and it stays phosphorylated. And it may even interact with the hormone-sensitive lipase. So here it is. These CGI58, when they are released from the perilipin, they may activate the ATGL that takes triacylglycerol, transforms that into the diacylglycerol and one fatty acid. Here you see the in this lipid droplet, here you see the HSL that takes these things and transforms that into a monoacylglycerol and two fatty acids. Here is MGL that deals with the monoacyl glycerols, and it splits glycerol and fatty acid. And then these fatty acids, they can be transported into the bloodstream, and then in the they can get to the tissues that need energy right now. And if, of course, if that tissue can if the tissue has some fatty acid transporter and it is able to catch that sort of fatty acids from the bloodstream, they are getting into the cells, and then in the cells they undergo so-called beta oxidation, and we will get to that mechanism just in a few moments, I hope. And that beta oxidation will give you ATP and produce some CO2, but the most interesting part is, of course, ATP, because ATP, that's basically energy that we use. So here is a small list of the events that are going in that lipid droplet. So I'm not getting into that because I have just shown you on this slide. Here is the entry of the glycerol into the glycolytic pathway. So we have fatty acids, we will do something with them, but we have a portion of the glycerol. Of course, we don't want to lose any of that, and we want to do something useful with the glycerol as well. So about 95% of the biologically available energy of the triacylglycerols resides in their long-chain fatty acids. So only 5% is contributed by the glycerol. And the glycerol released by lipase action is phosphorylated by glycerol kinase, and resulting glycerol 3-phosphate is oxidized to the dihydroxyacetone phosphate. And the glycolytic enzyme triose phosphate isomerase converts this compound to glyceraldehyde 3-phosphate, which can be oxidized via glycolysis. Here you see this scheme and the chemistry of the process. So here you see the glycerol. The glycerol kinase, it uses a bit of energy, but that transforms it into the L-glycerol 3-phosphate. So here it is. Once again, we have glycerol 3-phosphate dehydrogenase. We get this sort of chemical. So dehydrogenates, you might guess, here you see this double bond O, and here you see OH. So it has dehydrogenated, removed the hydrogen atom, and here you have this sort of double bond. And triose phosphate isomerase gives you this sort of diglyceraldehyde 3-phosphate that can go into the glycolysis pathway, and through the glycolysis it will get to the Krebs cycle, and in the Krebs cycle it will produce via the Krebs cycle and electron transport chain, it will produce a lot of ATP. Well, compared to the fatty acids, it's just as I told you, just 5%, but anyway, we don't want to lose even that energy, because why? So fatty acids, they are for that sort of processing and getting energy out of them. You remember that Krebs cycle, it's going on in the mitochondria, so we should get all these chemicals into the mitochondria to do something with them. And there is certain we need to transport somehow fatty acids. We need to activate them and transport them into mitochondria. The enzymes of fatty acid oxidation in animal cells, they are located in the mitochondrial matrix, discovered by Lehninger. So if you have ever tried to read some books on biochemistry, I'm sure you did, then you should probably know the famous book of Lehninger, and maybe some other books, but this one is pretty prominent, and that's, I guess, the same Lehninger. And the fatty acids with chain length of 12 or fewer carbons, they enter the mitochondria without the help of membrane transporter, so they can just diffuse. If you have 14 or more carbons, that's our majority of fatty acids, so typical length is something like 16. They need some help. This help is called the carnitine shuttle. That undergoes in 3 reactions, and the carnitine mediated entry process, that's a control point. So that's a point when you control how much energy do you need from that fatty acids. Here is the formulae of the carnitine. So here you see that on the right. Pretty interesting chemical, and I guess we can we can look at the reactions of this carnitine shuttle that we need to transport everything into the mitochondria. So 3 reactions, as I told you. So let's consider these 3 reactions. So first reaction of the carnitine shuttle. It is catalyzed by the family of isozymes of acyl-CoA synthase, each specific for fatty acids having either short, intermediate, or long carbon chains. So that depends on different carbon chains. And in medicine, there may be certain conditions connected to some defects in these enzymes. As you see, we have different enzymes that depend on the length of the fatty acids. So it may so happen that there are certain problems with fatty acids of certain lengths only. So some of them are doing pretty okay, while other they have different length, and they are not so okay. These isozymes are present in the outer mitochondrial membrane. Uh, Let me check. I should have yes. He or it's not. Um, so they should be present on the outer membrane. Let's check the reaction. Here we have what do we have here? So we have fatty acyl CoA synthase. That's the step. The carboxylate ion is adenylated by the ATP to form a fatty acyl adenylate and pyrophosphate, and the pyrophosphate is immediately hydrolyzed to molecules of inorganic phosphate. So here you see the fatty acid. These are that's long tail. Here you have ATP, and here you see the attack, the nucleophilic attack of these negatively charged oxygen on the phosphorus, and this part goes away. So that will be the pyrophosphate. And we have previously seen reactions that give up the pyrophosphate. That's a really common part in the biochemistry when you have certain reaction, and this reaction is almost in equilibrium, and it really helps if this reaction is coupled to making two inorganic phosphates from the pyrophosphate. That basically blocks the reverse pathway for that reaction. So you will see this sort of chemistry pretty often. Here is some sort of here are some sort of free energies for different reactions. Okay, but let's consider them one by one. So here we see the fatty acyl adenylated adenosine, and here you see the coenzyme A. Coenzyme A has this sort of SH group, and this non-bonding electron pair on the sulfur may attack the carbon atom. So you might guess once again a nucleophilic attack, because we have electronegative oxygens near this carbon atom, and this carbon atom has a little bit plus charge, so it can be attacked. And the sial group of this coenzyme A, it attacks the acyl adenylate and displaces AMP, forming the thioester fatty acyl CoA. So here it is. So that's the result. What's next? So now the second reaction that we have in this sort of Carnitine shuttle. So fatty acyl CoA esters formed on the cytosolic side of the outer mitochondrial membrane. They can be transported into the mitochondrion and oxidized to produce ATP, or they can be used in the cytosol to synthesize membrane lipids. So there are two fates. So we have these sort of this sort of reaction. We have acyl CoA, and then we can use this acyl CoA depending on what we want for either producing some sort of membrane lipids, for example, and adding to the cell membrane, maybe to some Golgi apparatus or endoplasmic reticulum, or whenever we need that. Or we can use them for pure energy and send to the mitochondria, get back carbon dioxide and a lot of ATP that can be used for different things. And the fatty acids that are destined for mitochondrial oxidation, and let's think we need energy at this point, they are transiently attached to the hydroxyl group of carnitine to form the fatty acyl carnitine. That's catalyzed by the carnitine acyl transferase 1. also called carnitine palmoyl transferase 1 CPT1. So here is CPT1. You see that's embedded in the membrane of the mitochondria. So here it takes acyl-CoA, releases CoA, and that CoA can be reused once again later. And here you see carnitine. That carnitine is attached to the fatty acid. The formula of the carnitine, here it was. So here you see the formula of the carnitine. So, and this thing, so what happens next? The acyl-CoA is converted to the carnitine ester as it passes through the outer membrane, so it goes through and it converts. Now, the fatty acyl-carnitine ester then diffuses across the intermembrane space and enters the matrix by passive transport through the acyl-carnitine cotransporter of the inner mitochondrial membrane. So here you see this sort of co-transporter. This thing, so we have connected our fatty acid to the carnitine and transported through the outer membrane. So you remember, mitochondria has two membranes. So I'm not going into many details, but here you see two membranes, here you see all the cristae, so intermembrane space, and here is the matrix of the mitochondria, so inside of that two membranes. So when it gets through that, it can just diffuse. There's not so big distance to diffuse. It gets to the transporter. That transporter exchanges one carnitine with the fatty acid attached for one carnitine without any fatty acid. So that's an anti-porter. And this basically moves the fatty acid carnitine inside into the matrix of the mitochondria. In the matrix of the mitochondria, this is the same image we had. I have just moved this sort of square to show you that we are now at the third stage, so the third reaction of the carnitine shuttle. That's the final step of the shuttle. You might guess that we have already transported everything inside. We just need to recover things to where they were at the beginning, and that's basically the fatty acyl group is transferred from carnitine to intramitochondrial coenzyme A by carnitine acyl transferase 2 called CPT2. So here is CPT2. This isozyme is located on the inner face of the inner mitochondrial membrane and regenerates fatty acyl CoA and releases it along with free carnitine in the matrix. Once again, a pretty common biochemical pattern. You have certain chemical on the outside of something. So I have some membrane. I can, and my chemical cannot pass through. So what's going on? This chemical is conjugated with something, so it's chemically connected to something that can pass through that membrane. It's connected. It passes through the membrane, and then it's disconnected. Pretty interesting thing may happen if you have a chemical that is constructed, let's say, from two parts. So I'm just drawing some schematics. So here we have a triangle and a circle, and we have some membrane that does not allow them to pass. But I want on the inside of this membrane, I want the same structure, so a triangle and a circle. And it may happen that I can somehow get a lot of triangles here, so I can either synthesize them or something like that. So I just need the circle to make them complete. So you will often see some biochemical pathways that are detaching this circle from the triangle, transporting it into the inside, and reattaching to another triangle that looks So all in all, if you take the overall sum of what has happened, that looks like transporting the whole chemical from the outside to the inside, but in practice you see that we have transported just this thing. Here you see even a more cunning way of doing that. When you have this sort of construct on the outside, so let's say a circle and a triangle connected together, and I have a different triangles on the inside, and I am detaching something from here, and I am anti-porting some triangles to the outside while this is transported into the inside. So there may be different, let's say, combinations. What I'm trying to show you that this sort of carnitine anti-porter, that's really interesting thing. So I'm transporting carnitine without anything, and I'm importing carnitine with something. So I can transport outside. I can transport something outside, some triangle outside, so he can pick up some circle, and then transport that inside, and on its place export once again some different triangle, and that looks like the circle was teleported to the inside. So there may be different interesting combinations when you are destroying your chemicals, then collecting everything together just for the sake of transporting them through different membranes. And you will see that through the biochemistry again and again and again. The same principle was when I showed you all that processing of the triacylglycerols in the intestine. So we have first destroyed everything up to the bits. So we had just monoacylglycerols and some fatty acids. We have uptaken that, then we have collected them together into the triacylglycerols. Then if we need, we may once again deconstruct, construct, so on and so forth. So depending on what we need to transport, where we need to transport, and so on. Okay. So the fatty acids of triacylglycerols, they furnish a large fraction of the oxidative energy in animals. So dietary triacylglycerols are, so like a reminder, they are emulsified in the small intestine by bile salts, hydrolyzed by intestinal lipases, absorbed by intestinal epithelial cells, reconverted into the triacylglycerols, and then formed into the chylomicrons by combination with different apolipoproteins. So that's what we have seen. These chylomicrons are deliver triacylglycerols to different tissues where lipoprotein lipase releases free fatty acids for entry into the cells. The triacylglycerols stored in adipose tissue are mobilized by hormone-sensitive triacylglycerol lipase, and the released fatty acids bind to the serum albumin. So once again, you might remember if you have ever performed any biochemical analysis. So serum albumin is one of the is on the list of what is commonly measured. So not only some levels of, I don't know, some hemoglobin and so on, but serum albumin is measured as well. And they are carried in the blood to the heart, skeletal muscles, and other tissues that use fatty acids as fuel. So, and once inside these the cells, fatty acids are activated to at the outer mitochondrial membrane by conversion to the fatty acyl-CoA thioesters, and these acyl-CoAs they enter mitochondria through the 3 steps of the carnitine shuttle, and then the oxidation begins. So here it is. It is called the beta oxidation. That's the beta oxidation pathway. And here you see what happens to all that fatty acids and how do they give us some energy. here on the right is the connection of beta oxidation and the Krebs cycle, or citric acid cycle if you wish. Here is the palmitoyl-CoA. It has 16 carbon atoms. Here you see it is connected to the CoA protein, so here it is. And here are the steps. Let me check if I have that in more. Well, I a bit have them, but not in very detail. Good. Let it be as it is. So here you see these steps. So first of all, the acyl-CoA dehydrogenase. So dehydrogenase means that it removes some hydrogens somewhere. It uses as a cofactor FAD, so flavin adenine dinucleotide. So here it is. I guess we have seen that when we have considered the electron transport chain. So we have seen NAD, NAD plus, NADH. I hope you remember that. So that was nicotine amide dinucleotide. And here you see FAD, FADH that we have met there as well. But this is FADH2, so it carries 2 hydrogens with it. And this acyl-CoA dehydrogenase creates you a double bond here. So it creates you a double bond by removing at alpha and beta carbons. It removes 1 hydrogen from each of them and creates you a double bond. That's basically trans delta 2 enoyl-CoA. So this delta 2 tells you that you have a double bond on the 1, 2 on the second carbon atom. Then we have a hydratase. This hydratase attaches water. So here you see the water attached and a forming of the OH group. So here it is. The water was attached to that double bond. Then we get through the beta hydroxyacyl-CoA dehydrogenase. This one uses NAD plus. So NAD plus to NADH. And here you see what happens. So we had this sort of OH group. Now we have this double bond. Now the acyl-CoA acyltransferase or thiolase takes 1 CoA atom and attaches it here. So here is a new CoA atom, and this part is now acetyl-CoA. Yeah, so that's the thing I asked you not to mix up with the acyl-CoA. So here you see the acyl-CoA. So acyl-CoA, long carbon chain. Here you see C14, yeah, so many carbon atoms. And here you see acetyl-CoA, just 2 carbon atoms. And these acetyl-CoA now can go to the Krebs cycle, and then some electrons will go to the electron transport chain. And at the end of the day, you will get some ATP from all of that machinery. So I guess we have considered the Krebs cycle, we have considered the electron transport chain, so you should know all that stuff already. But here is the fuel for that Krebs cycle that will be transformed into the energy in form of ATP for us. So here it is. And here is what is left, and you might guess that what is left is pretty similar. So many of these reactions, they are to create. So we not just split some bond here, but we are trying to create the same sort of head on the acyl part that will be attached to the CoA, so that we can return to the beginning and perform basically the same reactions that we already had. So we are trying to recreate our chemical just 2 carbons less. And if we are able to do that, if we are able to recreate that chemical, that's perfect, because we can now just go once again and again and again. And this is basically the scheme how it's going. So we get with C14 we get acyl-CoA for the Krebs cycle, and we are left with C12. With C12, we get, once again, we get C10, C8, C6, C4, and then C4 is split into two acyl-CoAs, acetyl-CoAs. So at each iteration of this sort of cycle, we can get one acetyl-CoA, and the remnant gets shorter by two carbon atoms. So that's basically the mechanism of the beta oxidation. So here you see how everything is going. So here you see FADH. It can be used in the respiratory chain. So NADH. So everything can be used, and is pretty useful for getting some energy. Here is example. Here is some example of chemistry that's called a retroglycan cleavage. So I wanted to consider a little bit more details of this of the mechanism of what is going on there, and how this reaction is performed. You see the overall reaction on the bottom. So here is basically some acyl-CoA. So at the last step of our of one iteration of the beta oxidation. So here that's this step. So here we had this sort of thing, and now we want to get this sort of thing. So we want to attach one more CoA, so one more coenzyme A, and transform this molecule into two molecules. So break this bond and attach their CoA. So we are considering right this step. And you see, so here is this step shown in some sort of schematics, and here is some detailed mechanism of the reaction. So it's a little bit more complicated than it looks like. So this thing, that's basically the coenzyme A. So it has SH group. This hydrogen is abstracted by some base, and these electrons, they may attack carbon atom. So we have once more some nucleophilic attack. They attack this carbon atom. Electrons from this double bond, they go to the oxygen. Now oxygen acquires certain negative charge. You see a pretty interesting structure here. Do you remember how is this structure? How I called that when we were considering all the chemistry and all that nucleophilic attacks? Do you remember what was this? I call that something intermediate. What kind of intermediate is this? Yep. Yes, tetrahedral intermediate. Absolutely correct. Yes, really good. You remember that. So here is a tetrahedral intermediate, and here we have this oxygen with some acquired negative charge. And then you certainly remember that some of these electrons will be used to reform the double bond, and one of the since carbon atom likes to have 4 bonds, not 5, you might guess that one of the groups will be rejected and will be a leaving groups. Will be a leaving group. So here you see this mechanism. So the bond is reformed. These electrons are used to create double bond here, while these electrons from this double bond, they once again go up to the oxygen, and it acquires some negative charge. So this is a sort of leaving group. Here you see the was it acyl. Well, well, this one part has left, so that's acetyl-CoA. I guess it has left, and now you see that once again we have certain reactions, like these electrons are recreating double bond here, these electrons are attacking some hydrogen and acquiring some new hydrogen, and this is the result that we get. Good. Of course, there may be certain caveats if we consider that fatty acids can be a little bit different. So they may have sometimes they may have some double bonds, and of course that will be not really good if our enzymes will be not capable of doing something with that double bonds. So we should have certain mechanisms to fix that problem. If there is a double bond, we need to do something with that. Moreover, we have considered the mechanism that takes basically two carbon atoms at a time. So we used two carbon atoms at a time. But what happens if our fatty acid contains some odd number of carbon atoms? So it is not divisible by 2, and we just cannot perform that operation and get to 0. So that should be so we should do something in this case as well. So here are different examples of what is happening in case we are in some of these cases. So let me check which one is which. I guess this one is the regular beta oxidation, so we have just some sort of creation of the double bond, we have hydration, we have once again oxidation and creating the double bond with the oxygen, and then we have dialysis and acyl and acetyl-CoA. So that's a simple case. Here I guess are first 3 stages of the palmitate degradation. So that's once again that's a simple case, but I just want to show you how it's going. So the first step, we are getting acetyl-CoA that contains these first 2 carbon atoms. Then we get acetyl-CoA that contains next 2 carbon atoms. So here they are in this image, and here they are here. And then the third iteration will give you these 2 carbon atoms. So here where they are I guess here they are, just a bit not drawn, and so on. So these are different iterations, and every time you get 2 carbon atoms. And here we have the cis delta 3 enoyl-CoA isomerase that allows continued beta oxidation of fatty acid with the single double bond. So here is palmitoyl-CoA. So here it is. We have a double bond here, so there is a double bond in the fatty acid. It was not introduced by our enzymes, it is already there. And what happens? We are performing beta oxidation as always for certain number of iteration, always removing 2 carbons at a time. So we have removed, but at some point we get to the situation where we have carbon atom attached to carbon atoms with double bonds. And that's a bit of a problem. So now we should do something with that. And here you see the cis delta 3 enoyl-CoA isomerase moves this bond from here to here. So here you see this double bond was moved. Did it get any better? And tell me why. No. Let's check the regular pathway that we had here on the left and look at these chemical. We have a double bond here. It's just situated in the right place. So now moving the double bond to here, we have basically recreated this thing. So yes, we came to this chemical through another pathway, but what do the enzymes care? So they see the chemical, they act. So it can undergo the hydration, oxidation, whatever is needed, and get to the acetyl-CoA and some remainder. So this is pretty reasonable way. So if we have a double bond somewhere where we don't like this double bond, it can be moved by these sort of cis-3-enoyl-CoA isomerase, and we get what we need. Let me check for a moment how many slides do I still have. So where are we? Okay, that's vitamin B. I guess some parts can be skipped. Okay, let me show you just few more formulae and we'll be at a good place to stop. Here is a oxidation of the lineoyl-CoA. That's a complete oxidation of the diunsaturated fatty acid. So you see two double bonds here. So now we should do something with the two double bonds. Well, if they are pretty far apart, it's not a problem. You can use the technique from the previous slide. But if these double bonds are pretty close, well, some other technique should be employed. And here you see basically what happens with that technique. So once again, cis-3-enoyl-CoA isomerase moved these double bonds to here. So here we have a new double bond. One acetyl-CoA can be removed, then acyl-CoA, and then that goes through the pathway recreating double bond here. Once again, trans delta 3 anoyl getting through dianyl-CoA reductase from these two double bonds, getting just one double bond here, and then through C3 delta anoyl-CoA isomerase, moving that bond to here, where it's convenient to perform the rest of the reactions. So pretty neat mechanism. The odd chain fatty acids, they yield so-called propionyl-CoA in the final step of the thiolysis. So if you have even number of carbon atoms, you can always separate them by 2 carbons, and you will always get acetyl-CoA. In this case, you perform everything, perform until you get to 3 carbon atoms. That is called the propionyl-CoA. With the propionyl-CoA carboxylase that is biotin dependent, you can get to the d-methylmalonyl-CoA. So here it is. Then it can be through the racemase, it can be transformed into the L-methylmalonyl-CoA, and then through the B12 dependent rearrangement, B12, that's vitamin B12, it is transformed into the succinyl-CoA that can be used in the Krebs cycle. So that basically allows for the entrance of that odd chain fatty acids into the Krebs cycle. Here are just few slides regarding the cobalamin dependent enzymes. This cobalamin, that's basically the coenzyme B12. So here you see the formula, looks pretty complicated. So here you see some quarrying ring. Here you see the benzimidazole. This benzimidazole is connected to the cobalt in here, and here you see different variations. So you may have so-called cyancobalamin. I guess that's the form that we are taking as vitamin B12 if I don't mix up things. You may have methylcobalamin, or you might have this sort of form. So it works well. There are certain reaction types that are catalyzed with these sort of enzymes. It's pretty useful, let's say, because we have few reactions of intramolecular rearrangement, some methylation reactions, some reduction of ribonucleotides to desoxyribonucleotides. So you might guess we need that to create our DNA. And these B12 reactions in mammals, there are only two of them, like L-methylmalonyl-CoA to succinyl-CoA and homocysteine to methionine. So methionine synthase that provides methionine needed for as adenylyl methionine and other coenzymes used in purine and thymine synthesis, and that are critical for DNA and RNA synthesis. So B12 is pretty critical for us. So here are just some schemes of the reactions that are catalyzed with this sort of complicated system. So here you see the chlorine ring, here is the cobalt. So pretty interesting that we have this sort of metal in our bodies, and moreover we desperately need that, otherwise we will have certain problems. And here is the scheme of the reaction. So the cobalt and carbon bond undergoes homolytic cleavage, yielding cobalt 2 plus and the 5' deoxyadenylyl free radical. So pretty rare thing to see. So here you see a radical reaction. So we almost all biochemical reactions were heterolytic cleavages. If we have some, well, they were heterolytic, and here we see the homolytic cleavage. That's pretty rare, but that's what we have. So the radical is converted to the 5' deoxyadenosine by abstraction of a hydrogen atom from the substrate and producing a substrate radical. So hydrogen is removed by electrons with one electron, and the other stays, and we have a substrate radical. The substrate radical is rearranged, producing a new radical with the carbon skeleton of the product. So you remember that carbon skeletons may somehow rearrange to get to a more stable state. We have considered carbanion and carbocation rearrangements where a hydride shift may be performed, or even some groups may shift. And here you see this is used to get some sort of result, and then we have this product-like radical. We have deoxyadenosine, and here is our cobalt, and we return this hydrogen to the product-like radical, and we get our product. And the hydrogen from the 5' CH3 of the deoxyadenosine is returned to the product-like radical to form that sort of product. And here you see once again that we have this sort of structure, and the bond between 5' and CH2 of the deoxyadenosine radical and cobalt is reformed, regenerating the B12 cofactor in its stable cobalt 3 plus form, ready to undergo another reaction. So that's a cycle that is performed, and this cycle is performed through a radical. So here you see this sort of scheme. So that's the bond that is being broken. It is homolytically broken, so that you get a radical, and this cobalt gets from 3 plus gets to 2 plus. Then it exists in this form. Then this radical, basically, as you see on this picture, it attacks the substrate, abstracts hydrogen, but abstracts that homolytically. So there is a radical left, a radical undergoes rearrangement, and then this hydrogen is returned to a more stable form of the substrate that is now a product. So that's what we see here. Here is just few images. I just want to get through all of these so that we don't have get through that tomorrow. Maybe I will stop. Well, yes, I guess yes, I will stop after the enzyme just to finish the idea. So here you see our cobalt 3 plus. Here you see how the radical was formed. So here you see cobalt, 1 electron cobalt got for himself. So now it's 2 plus, and 1 is left here, so this is a radical. So here is a cobalamin cobalt 2 plus form. Here you see some rearrangement, and here is the reaction that is crucial for us. That's L-methylmalonyl CoA. Here you see the 5' deoxyadenosine. That's basically that's this part. So this part that was transformed into the radical. So here it is. It has abstracted hydrogen from here, so here was the hydrogen, and this was in the form of a radical. Now it has taken that hydrogen, and now the substrate is in the form of a radical. Now here we have a rearrangement. So here you see this part moved to here. So now this is the radical. The logic behind these shifts is the stability. So how good can you, let's say, delocalize some charge in the molecule, and in case of having only hydrogens, you don't have a lot of ability to delocalize that charge. In this case, you see carbon atom that is connected to another 2 carbon atoms, so there is much more ability to delocalize some charges and to be more stable. So that was pretty reasonable transformation. And now this more stable form may get back its hydrogen from the 5' deoxyadenosine, so it gets this hydrogen back. And here you see once again the radical was reformed, and here we have our product, so succinyl CoA. So that succinyl CoA we have created from the malonyl CoA that we needed. So where it started, we had some odd chain fatty acid. That odd chain fatty acid yielded a propionyl CoA, because we cannot just deconstruct it into the parts with 2 carbon atoms. This propionyl CoA was transformed into the D-methylmalonyl CoA, that is then transformed through some steps into the succinyl CoA that can enter the Krebs cycle and be transformed into energy for us. So that's a way to deal with this sort of thing. And how that was performed, these scary looking formula, so this scary looking chemical transformed part of it, like this top part, into a radical. That radical abstracted some hydrogen. The molecule that lost its hydrogen transformed to get more stable. Then it got back its hydrogen, and we obtained our product. And this thing was reattached to the cobalt, and this sort of cycle returned to where it was. So that's basically the logic of what has happened. And here you see the active site of the methylmalonyl CoA mutase. So notice that a histidine residue from the enzyme binds to cobalt in place of the benthimidazole. So this arrangement of substrate and coenzyme in the active site facilitates the cleavage of the cobalt carbon atom bond and the subsequent abstraction of the hydrogen atom of the substrate. So here you see that's part of the histidine. So this is the cobalt atom. So here is the cobalt atom. Here is deoxyadenosine that will be transformed into the radical. So here is this part. And here you see a displaced benzimidazole. So this part was just moved aside, and then cobalt was attached to the histidine, and that helps a little bit to facilitate the reaction. In the scary looking formula, this was the part. So this part in this scary looking formula, so here you see a connection to cobalt. In the last slide in here, that's this part, and now it has lost its connection to the cobalt, so this was previously connected to this thing, but now it's connected to histidine. So I hope you can guess how that looks in 3D. Good. What's interesting, this methylmalonic acid has some clinical use. So why do we use that? Some serum levels of B12, they may be unreliable because there are some inactive forms that may inflate result. So in case of deficiency of B12, it's more reliable to measure the methylmalonic acid. And methylmalonic acid rises only when B12 dependent methylmalonyl CoA mutase fails, and that's the best marker of functional B12 deficiency. So that's really good. We have only 2 reactions that need that B12, so it's not so large pool of reactions, and we can check whether they go as planned, so whether we have sufficient amount of the B12 vitamin in the organism. If we try to measure that as it is, well, that may be a little bit unreliable because we may measure something that is in some inactive things. While this MMA, if it rises, that's definitely the sign of something went wrong. So it's early, it's sensitive. It may give you some false positives, of course, nothing perfect, especially in renal failure, and it's often used in newborns to screen for metabolic disorders. And here are some references for you. So borderline levels for B12 like 200 to 400 picogram per milliliter. So if we have some problems, they may cause some neuropathy or even cognitive decline. And of course there should be some differentiating B12 from other kinds of deficiencies. Here you have some just overall table of how much energy do we get from beta oxidation. That's just to look you for you a little bit later. And no, we will not get to this, but I will show you a bear. So you know that bears, they can hibernate for a pretty long time, so up to 7 months of continuous dormancy. Their body temperature is approximately 31 degrees C during this time, so that's pretty close to normal. And they use from their fat stores something like 25,000 kilojoule per day, and they are not eating anything, drinking, or so on. And of course there are some physiological changes, like heart rate drops just impossibly low, and respiration drops very low. But the interesting part is that they use body fat exclusively, and the fat oxidation provides enough energy and water and substrates from gluconeogenesis, so they can survive through the winter. So that's really interesting. There are some more interesting notes here, so you may look at them later, but that's a really interesting process, and we are pretty interested in this sort of hibernation because it has some medical and scientific relevance, because the insights may help us by preserve, let's say, organs for transplantations, or maybe they will have some applications in human torpor for, let's say, long duration space missions. So why not? So send someone to Mars and let him sleep for 3 months, and then he wakes up and he's where he should be. Okay, so I guess with this nice looking bear we can end our lecture. if you have any questions or comments so far, please fire away. Okay, no comments, no questions. Good, then thank you for your attention. Have a nice evening and see you tomorrow.